Welcome to Dr. B.R. Ambedkar Open University. Today we are discussing the BSc first year UG first sem. Main topic is the viruses replication. Today we discuss the what are the viruses. The viruses are the non-living things. The first one is the Ivanovsky, the first discovered in the Ivanovsky. The name is given by the Bering. What is the meaning of the viruses? Viruses coming from the Latin word, mainly meaning of the poisonous. Another is the disease causing agent is also called as the Contagium vivum fluidum. Mainly viruses are there containing some of the nucleic acid, either RNA or DNA. Most of the plant viruses are there containing the RNA. And some are the animal viruses and the bacterial viruses are there containing the DNA. Some are the exception conditions. Cauliflower mosaic viruses, these are the plant RNA, plant viruses, mainly these are the containing the DNA, these are the exception condition. Next one, some are the influenza viruses, F2 choliophase also, these are the animal viruses, these are the animal viruses. At the same time, these are the containing the RNA. The main plant viruses are there containing the RNA, animal viruses are there containing the DNA. Next one is the bacteriophase. What is the meaning of the bacteriophase? Bacteriophase mean some are the viruses are attacked by the bacteria. Okay, for example, F2 bacteriophase. Next one, most of the even phases of the bacteriophase, T2, T4, T6. Mainly the bacteriophase, they are containing the double stranded DNA. Especially they can divide the bacteria into two types. Mainly two parts is there. One is the head, and next one is the tail. Head is that they are containing the double stranded DNA. Tail is that they are containing the some are the tails, these are the fibrils. The head is the especially they are double stranded DNA. Tails are the especially 6 tails are present in bacteriophase. Prion and virion. What is the meaning of the virion? Virion mean their nucleic acid plus next one is the protein capsid. What is the meaning of the protein capsid? Especially the outer structure of the viruses that they are containing the protein capsid. These are the mainly they can form the number of components is available in protein capsid. And next one is the nucleic acid, either DNA or RNA. The both are they cannot see in the one viruses, only they can either RNA or DNA they are containing the some are the viruses. Next one, these are the virion. Next one, virioid. Virioid is the only nucleic acid components. Okay, there is no protein code and then there is no capsid. Okay, main capsid mean outer layer of the protein code. Okay, and virioid is the only nucleic acid. The example of the virioid is the next potato spindle tuber viruses. Or mainly these are the only virioid. And next one, replication of the viruses. Replication of the viruses, they can see two types of the cycles are they can see in the replication of the viruses: lytic cycle and lysogenic cycle. Mainly lytic cycle is the mainly they can see in the T even phase are attacked by the some are the E. coli bacteria. T even phase. What are the even phases? T2, T4, T6, etc. Next one, lytic phases are the mainly they are containing the 5 steps. One is the absorption, next one entry or penetration, and next one is the transpiration, and next one is the synthesis, next one biosynthesis formation, next one assembly, the finally is the lysis. These are the steps are involved in the lytic phase of the replication of the viruses. What is the meaning of the mainly what are the lytic and lysogenic cells? These are the mainly viral multiplication. Lytic cycle also mainly lytic cycle and the lysogenic cycle, the both are the important in the replication of the viruses. What are the difference between the lytic and lysogenic cycles? These are the both are the they can see in the viruses life cycle, lytic cycle and lytic cycle, lysogenic cycle. Lytic cycle also mainly DNA of the viruses does not integrate in the host DNA. These are the very important. Does not integrate in the host DNA. What are the host? Especially E. coli bacteria are the host. These are the mainly these are the bacteriophage affected or interacted or affected by the attack by the DNA, E. coli bacteria. These are the E. coli bacteria mainly. These are the host cells. Mainly the DNA they cannot integrate into the host cells. Lysogenic cycle is the integrate into the host DNA. Mainly integrate, mingling. Integrate means mingling of the host DNA. Actually, bacteriophage are the double stranded DNA. They are containing the double stranded DNA, and the bacteria are they are containing the single stranded DNA. Okay. Next one. Host DNA is hydrolyzed in lytocycles. Is the host DNA is the hydrolyzed. What is the meaning of the hydrolyzed? Hydrolyzed means especially the dissolving part, especially the bacteria cell wall membrane, especially they contain the muramic acid, peptidoglycan, different types of the muramic acid, gram positive bacteria, gram negative bacteria, different types of the bacteria, mainly the composition of the muramic acid. Mainly these are the composition of the muramic acid, the hydrolyzed by the lytic cycle. Mainly the host DNA hydrolyzed. What are the host DNA? Host DNA, the double stranded DNA is the hydrolyzed in lytic cycle. The host DNA they cannot hydrolyzed in lysogenic cycle. Next one, propase also available in the lysogenic cycle. The absence of the lytic cycle. Next one, DNA replication mainly then independently from the host DNA replication in lytogenic cycle. Lytic cycle. In the lysogenic cycle as there is no independent. These are the depend on the mainly they are depend along with the host DNA replication. Next one is the mainly it occurs into the very short period. What are the short period? Approximately 30 minutes to 90 minutes are the lytic cycle. In the lysogenic cycle is the takes over time approximately more than 1 hour. Okay. Next one is the symptoms of the viral replicant are evident. Is the symptoms of the viral replicants are the evident in the lytogenic cycle. Symptoms of the viral replicate not evident in lysogenic cycle. Mainly genetic recombination also. Genetic recombination is not allowed in the lytic cycle. Lysogenic cycle the genetic recombination is the allowed into the in lysogenic cycle. Totally the undertaken by the viral genome by the in lytic cycle. These are the distributed for the viral genome in lysogenic cycle. Okay, these are the major difference between the lytic cycle and lysogenic cycle. Okay, and next one is the steps are involved in the lytic cycle. Absorption. What is the meaning of the absorption? Absorption process especially mainly actually the bacteriophage is the head and tail structure. Head is they are contain the double stranded DNA. The tail is they are contain the how many fibrils are there contain? 6 fibrils. Mainly are the 6 fibril attached by the outer structure of the bacterial membrane. These are the reversible action. Actually, these are the attached by the reversible action, the bacteriophage. See actually the absorption facilitated the negatively charged carboxylic group on the host surface and the positively charged amino group of protein present in the tip of the phase tail. Actually, tip of the phase tail is especially they are containing the 6 fibrils. Mainly this is the absorption. This is the attached by the some are the bacteriophage attached by the bacteria. Especially we are studying the E. coli bacteria. Okay. Next one is the already we in T even phases are the first one fiber first attached the cell surface. Mainly they can finally first one is the attached by the cell surface. What are the cell surface? Actually the bacteriophage the outer structure is made of the peptidoglycan, muramic acid. These are the hardly nature. Okay, these are the fibrils are attached by the outer surface cell surface. Next one, penetration or is also called as the entry or infection. Mainly penetration or entry. Mainly bacteriophages are the enter into the in host cells. Mainly after absorption of the particle secretes in the enzyme. Mainly some are the enzymes. Already we discussed the hydrolyzed. Host cells are the hydrolyzed in lytocycle cycle. In lysozyl cells, the host cells they cannot hydrolyzed. In the hydrolytic cycle mainly, some are the bacteriophage are the they can secrete some are the enzymes. These are the enzymes are secret. The outer structure is the hydrolyzed. Okay, hydrolyzed mainly the bacterial cells are the already contain the muramic acid complexes. Okay, and next one these are the fibrils are the enter into the pore also they can form to the outer structure of the bacteria. Okay, these are the four. Next one is the tail also they contract the some are the central tubular part is the actually is the core part and the tail part. Core part are also available in the double stranded DNA, and next one is the tail part is the fibrils. Actually the double stranded DNA they enter into the in bacteria. The empty of the bacteria head part is also called as the ghost. Okay, the empty protein shell of the phase is also called as the ghost. The ghost cells already double stranded DNA they can enter into the in E. coli bacteria. Okay, these are also called as entry. Okay, entry is also entry. Okay, next one is these are the structural diagram of the for lytocycle cycles. Actually these are the number of phases number of cycles are the available. Absorption, next one is the entry, next one synthesis, next one is the biosynthesis, assembly and lysis. The finally the cycle is also lysis cycle. Okay, these are the number of the different types of the steps also involved in the replication of the viruses, especially in lytic cycle. This is also the viral attachment of the in cells are the mainly. The next one is the outer structure uncoating. Next one is the enter into the nucleus. Actually the bacteria they are containing the only single stranded DNA. In the bacteriophage also they are containing the double stranded DNA. Double stranded DNA, the single stranded DNA is also called as the plasmids. What are the plasmids? The replicant part. See this one, the fourth one is the genome replication. Genome replication, especially the genome replication is the related to the bacteria. And the next one is the ghost and the entry of the bacteriophage in enter into the they can form the some are the double stranded DNA. These are the double stranded DNA. They can convert the mRNAs. Okay, this is the one type of the especially structural lytosine cycle. Next one. And at the same time some involved in the Golgi bodies, different ribosomes. Mainly the ribosomes also available in the inter part. What type of the ribosomes are present in the bacteria? 70S ribosomes. Already bacteria is the eukaryote. These are the containing the 70S ribosomes. Bacteria is the prokaryotes. And the next one is the most of the eukaryotes that containing the 80S ribosomes, and next one is the prokaryotes are that containing the 70S ribosomes. Okay, see the 70S two types of the subunits. One is the 60 plus 30 subunits. Next one is the different types of subunits in the ribosomes. Okay, is also involved in the ribosomes and translocation. Next one is the transcription and the synthesis, main formation. The first step is the adsorption, entry. The third step is the lytogenic cycle is the transcription or synthesis, mainly synthesis, formation. Okay, synthesis. At the same time phase nucleic acid takes enter into the inside of the bacterial cells, mainly synthesis of the proteins of the phase particles. Actually the bacteria inner part of the bacteria they are containing the single stranded DNA is also called as the plasmid. Okay, single stranded DNA. These are the replicate. Okay, the DNA phase replicates these are the actually the single strand is the conservative process. Mainly single strand is also called as the plasmid. Majority of the DNA act as the template. Okay, the single stranded DNA is also act as the template. Template mean mainly they can form the single form of the DNA. Okay, mainly synthesis. In synthesis form template synthesis viral specific of the mRNA by utilizing the mainly RNA polymerase. What type of the enzymes are involved in the synthesis process? RNA polymerase enzyme they can involved in the viral synthesis. Mainly mRNA they can convert the mainly these are the DNA, the single strand template they can convert the mRNA by the RNA polymerase an enzyme. Newly formed mRNA direct the host cell to synthesize the proteins which are used to the build up the some of the protein caps, especially protein caps, different types of the protein caps also available. Most of almost end of the replication phases, nucleic acid in the protein phase, lysozyme. The another important enzyme is the lysozyme is also important. What are the importance of the lysozyme also? They can involve the especially the single stranded DNA, they can convert the RNA polymerase enzyme. At the same time lysozyme enzyme also they can involve in the both the process also in the synthesis process, synthesis and replication. Next one, fourth process also the biosynthesis or the formation of new phase particle. Already in third phase the synthesis process also they can form the mainly template. The template or they can convert the already mRNA in the what type of the enzyme involved in the RNA polymerase enzyme. RNA polymerase enzyme, the mRNA, they can form the assembling. The process is also called the maturation. Is also called as the maturation. These are the mainly controlled by the viral genome. What are the viral genome? Double stranded DNA. The double stranded in the bacteriophage are they contain the double stranded DNA. The double stranded DNA, these are the mainly controlled by the viral genome. Mainly the maturation process are the controlled by the viral genome. Mainly viral genome are the double stranded DNA. They are the mainly the process are initially condensation nucleic acid molecules are that take place. The fourth process mainly the most of the in main process are the formation. Formation is the stages at the tail and the next one is the biosynthesis. Mainly the biosynthesis mRNA polymerase enzyme. mRNA polymerase enzyme they can involved in the biosynthesis process. Next one assembly. Mainly these are the assembly, actually these are the 200 phase development of the 200 phase are the formed within the takes place 30 to 90 seconds. Already we discussed the lytic cycle are the approximately 30 to 90 seconds are we taken the lytic cycle. The lysogenic cycle are the mainly they can see in the approximately more than 1 hour. Okay, mainly are the they can secrete the isozyme enzyme. The enzymes especially they can affect the host cell wall. What are the host? Host is the bacteria. Mainly are the host are the bacteria. Mainly are the bacteria are mainly outer structure is the they can made the muramic acid. The host cells are the mainly the cell wall and the phase particles also liberated. Mainly assembling they can form the group of forming the bacteriophages. They can form the group of bacteriophages. Next one mainly they can involve in the assembly process. They can number of mainly the assembly process they can control by the some are the E. coli repressors and they can form the theta like in the structure. Okay, mainly the DNA is the converted in the circular DNA. Actually the most of the bacteriophage double stranded DNA they can convert the single stranded DNA. Already bacteria they are containing the single stranded DNA. The single stranded DNA is the mainly the single double stranded DNA also they can convert the single stranded DNA, template DNA. All are they can form the assembling form. Mainly these are the assembling form, especially they can see in the fourth stage maturation genome is the wrapped in the protein coat, the assembling the number of assembling form they can see in the assembling form. Next one is the lysis. Lysis mean mainly lysis are the already see this one is the lysis process. Okay, the lysis of the bacterial cell and the release of the some are the virions. Already we discussed the virions. What is the meaning of the virion? Virion mean they contain the nucleic acid either the DNA or RNA. At the same time plus protein capsid, the outer membrane. These are the lysis process. The total process of the DNA replication is especially the lytic cycle in especially T even phase of some are the bacteriophage. Adsorption, entry, and next one is the synthesis, biosynthesis or maturation. Okay, assembling. Okay, biosynthesis is also called as the maturation. Next one is the assembling. Assembling mean they can number of bacteriophage also they can see in inner structure of the bacteriophage. The finally they can lysis. Lysis is the very important. The lysis, the availability mainly some are especially virus are the availability of the host they can living things. Mainly are the non-living things. Availability of the organism and availability of the host they can living the particles also seen in the lytic cycle. The next one is the lysogenic cycle. Lysozyme is also mainly number of steps also in one: attachment, entry, integration, and cell division. Is also lysogenic also mainly lysogenic. Mainly are they cannot host hydrolyzed. Okay, host hydrolyzed in the lytic cycle. Host they cannot hydrolyzed. Hydrolyzed mean especially some are the enzymes are already released in the lysozyme enzyme, RNA polymerase enzyme. Different types of the enzymes are involved, released in the outer structure. They can the dissolved condition. Host DNA hydrolyzed in RNA, especially lytic cycle. In lysogenic cycle, attachment. Attachment mean the bacteriophage or the lysogenic also they can attach to the some of the bacteria. Actually bacteriophage are the attack to the E. coli bacteria, attached by the E. coli bacteria. Actually bacteriophage mean mainly some are the viruses attacked by the bacteria is also called as the bacteriophage. Okay, in lysogenic cycle also attachment mean attach to the outer surface of the cell membrane, outer surface of the host cell membrane. Okay, entry also already we discussed the in lysogenic cycle. They can form the entry, especially the fibrils are the injected to the internal part of the bacteria. Mainly what type of the injected? Mainly the bacteria double stranded DNA are injected into the host cells. At the same time, in lysogenic cycle also bacteriophage inject to the DNA bacterial cells. Mainly the DNA are injected in the internal host cells. Okay, integration mainly entry, attach and absorption, entry, integration also mainly they can form the assembling DNA, form DNA molecules. Next one, double stranded DNA, they can convert the single stranded DNA. Mainly the single stranded DNA they can form the mainly RNA polymerase. Okay, these are the mainly the two RNA polymerase lysozyme enzymes are all are in the integration. Integration means the assembling process also they can see in the lysogenic cycle. Next one, cell division number of, especially in cell division process, approximately number of bacteriophage they can form in cell division. Okay, next one is the actually bacteriophage, especially bacteriophage are they can discovered by the twat and DRLE. Okay, twat and DRLE, and some at the same time some are the TMV, tobacco mosaic viruses. These are the crystallized by the W. Stanley in 1935. He got the Nobel Prize in 1944. Okay, actually what is the mainly Stanley? Mainly they can crystallized form. Crystallized means they can number of some are the chemical components they can form the crystallized form. Mainly TMV, tobacco mosaic viruses, they are containing the some of the RNA particles. Next one is the some are the capsomeres, internal part of the some of the proteins, different types of the protein. Mainly the replication of viruses or lysogenic cycles is the mainly developed by the discovered by the A.L. Loff. Mainly these are the also called as the temperate phase life cycle. The first one, these are the adsorption phase also they can available in the lysozyme. Adsorption, entry, next one is the recombination, and next one is the cell division. These are the steps are involved in the lysogenic cycle. Next one is also is the lysogenic cycle of the bacteriophage. What are the absorption? Mainly absorption in the bacteriophage isogen strains enter into the outer structure, especially outer surface they can form the adsorption. Next one, entry are the especially in the empty form also called as the ghost. The ghost are the empty form, the double stranded DNA they can enter into the in bacteria. Okay, in the bacterial cells. And the next one is the prophase. The especially prophase is the very important. Prophase phase is absent in lytic cycle. In lysogenic cycle is the prophase is the present. Is the major difference between the lytic cycle and lysogenic cycle. Okay, the prophase also they can see in the internal structure of the bacteria. Integration of the viral DNA. Okay, in cell division, what are the cell division? In cell division process they can form the two form of the bacteriophage. Okay, as actually in lytocycle cycle there is no cell division. Okay, in the cell division they can form the two, actually one cell they can divide the two cells. The two cell process also they can see in in lysogenic cycle. The cycle they cannot see in the cell division, only they can the assembling form. Number of bacteriophage are available in assembling form. In cell division also they can see in lysogenic cycle. Next one is the viral genome as well released, and the next one is the lysis of the mainly bacterial cells are the bacterial cells lysis. Also these are the lysogenic cycle and lysogenic cycle. Okay, these are the totally viruses mainly. Okay, mainly viruses are they can cause the number of diseases, especially the plant viruses, mainly transmission by the insects. Next one, aphids, nematodes and mites, especially especially the number actually the cell membrane of the viruses are the easily attacked by the some of the insects also, aphids, mites, nematodes. Next one is the hoppers. Okay, mainly insects are the vector group of the plant transmission group, mainly transmission groups. Next one is the some are the viral disease in plants. Okay, some are the viral disease, tobacco mosaic viruses, mainly these are the mosaic mean especially the number of group of cells they can form the mosaic form. Okay, in mosaic cells. Okay, especially mosaic TMB mosaic viruses firstly is the discovered by the W. Stanley in 1935. Mainly these are the they can see in the some are the tobacco family. It's the tobacco family, tobacco plant especially belongs to the Solanaceae family. Tobacco affected by the TMB tobacco mosaic viruses. Next one is the cauliflower mosaic viruses. Already we discussed the cauliflower mosaic viruses. Mainly they are containing the actually most of the plant viruses are they are containing the RNA. Some exception condition, cauliflower mosaic viruses they are containing the DNA. Okay, some are the viral disease. Next one, barley yellow dwarf, bud blight, sugarcane mosaic viruses, lettuce mosaic viruses, leaf curl viruses, papaya leaf curl viruses. Okay, next one is the some are the viral diseases of animals. In the recent days they can see in the lumpkin skin disease. Lumpkin skin disease also they can affect the in most of the cows, especially buffaloes, mainly are the attacked by some of the viruses. Lumpkin skin disease also. Next one, foot and mouth disease also affected by the some of the viruses. Next one is the pest disease. At the same time, AIDS. Okay, next one is the some are the rhinoviruses, influenza. Next one some are the sneezing. Okay, blue ear disease also. The rabies, especially African swine flu, mainly African swine flu, avian influenza and fluid mosaic. The fluid especially avian influenza especially they can cause the bird flu. Bird flu is the mainly they can affect the number of birds especially. And the Rift Valley fever viruses and sheep. The hepatitis C also they can number of viruses. The final conclusion of the viruses replication. The final conclusion of the viruses replication also, viruses mean poisonous, these are mainly affected by the disease the vivum contagium fluidium. Okay, these are the mainly first one is ionovisci, next one next one is the Bezering. The name is given by the Bezering, T.W. Stanley and mosaic virus. Mainly in the replication of the viruses, they can main involve the two life cycles, lytic cycle and the lysogenic cycle. Lytic cycle also they can involve the so many steps also involve absorption, entry. Next one is the entry also called as the penetration. Next one is the biosynthesis, synthesis formation, assembly and lysis. The number of steps also involved in lytic cycle. And next one is lysogenic cycle also, mainly they can see in the adsorption, entry. Next one is the mainly cell division and recombination. These are the steps are involved in the lysogenic. Mainly the recombinant DNA technology they cannot see in lysogenic cycle. In the recombinant DNA molecules they can see in lysogenic cycle. Another important is the host DNA is idolized. Host DNA is idolized in the lytic cycle. The host DNA they cannot idolize in the lysogenic cycle. Mainly, okay, number of mainly these are the virus are the mainly they can see in what main important is the availability of the organism. Availability of the host organism they can live in in internal part. Okay, main important theme is the replication of the virus. They can form the number of replicant, number of templates also, most of the T interferon also, interferons, most of the nowadays they can use for the interferons. These are the interferons also, mainly they can use for the some are the vaccines, mainly they can produce the vaccine, corona vaccine, different types of the vaccine, smallpox vaccine, rabies vaccine, polio vaccines, different types of the vaccine. These are the mainly mainly the main part of the replication of the viruses. These are the viruses. Okay, thank you.